This is going to be another video which doesn't have a script, where I want to talk a little bit about the process personally of me coming to the point where I felt like it was safe to get the new coronavirus vaccines. Now, I admit, when I heard of the Moderna vaccine being developed and this being the front candidate picked by the NIH for vaccination, I was very skeptical because mRNA vaccines had not been developed for widespread public use at that point. I am a big vaccine advocate in my infectious disease field. I work primarily with HIV positive patients in the outpatient setting, and prevention through vaccination is a large part of my treatment strategy. So I'm definitely in the pro-vaccine camp, but I was a bit skeptical because mRNA vaccines had not been really proven on a wide scale at that point. My problem, though, was my doubts were really unfounded when I actually did the research and the reading required. At that point, I felt much better about the mRNA vaccines. And in learning about how they work and how they were developed really busts a lot of the myths surrounding the vaccines. So first of all, mRNA vaccines are not a new thing. They have been developed for other conditions like Zika virus. But the nice thing about an mRNA vaccine is that it doesn't require the actual virus to be injected into someone, which could potentially cause side effects. So some viral vaccines are what they call a live attenuated virus vaccine. The nice thing about a live attenuated virus vaccine is it provides a very robust immune response, but at the same time, it is a very weakened version of the virus, but it can still cause some of the virus side effects in people. A good example of this was the old vaccine that we had for shingles, which was called Zostavax. And this particular vaccine was a weakened version of the varicella zoster virus. But even in a weakened state, if you give it to someone that was immunocompromised, like someone with active HIV virus or AIDS, it could trigger them to get shingles. The mRNA vaccines do not have this problem because, myth number one, they are not the virus. The mRNA vaccines encode for a protein that's found on the surface of the virus. That protein is the spike protein, which is responsible for its recognition of the cells that the virus can infect and also its docking and entry into the cell. So if you can make antibodies that block that spike protein, the virus doesn't get into the cell. But the mRNA vaccine is so much more than just forming antibodies. That's just a tiny part of it. And I'm going to try to explain the immunology of this completely off the cuff with no script in a way that kind of makes sense. Warning. I will be spending the next 12 minutes explaining the mechanism of action and immunology of the COVID vaccines, because the vaccine is so much more than get shot, get antibodies. I will show you exactly how the vaccine generates immunity to coronavirus. I believe that understanding this detail allows one to naturally dispel the myths surrounding the vaccine, understand why vaccine side effects occur, why the vaccine takes several weeks to become effective, and appreciate the beauty of the technical accomplishment of the scientific community that developed this vaccine. It's going to be a lot of information, but reduced to the simplest way that doesn't leave anything major in how the vaccine works out. I don't recommend doing this, but if you want to skip this section, fast forward to about minute 17 of this video. So, part of the problem with developing an mRNA vaccine before this was keeping it stable in the person long enough for it to have an effect, because the mRNA breaks down quickly inside the body. This again addresses another myth about the vaccine, in that the vaccine stays inside the body for a very long time, and that could not be further from the truth. Once the mRNA is made into the spike protein in the cells, the mRNA in the vaccine is degraded, gone, bye-bye. It doesn't replicate. So that myth is completely busted. But in its development in recent years, they found a couple ways to make the vaccine more stable inside the body, one of which was in the processing of it. They put it basically in a fatty bubble that almost looks like your own cell membranes to keep it from getting attacked or broken down inside the body before it gets to the cells. So once it gets into your cells after it's injected, it does not go into the nucleus. It has no way to get into the nucleus. So the nucleus is what contains your DNA. So there's a lot of people that think that the vaccine is going to alter your DNA. The mRNA vaccine has no ability to get inside the nucleus. So there is absolutely 0% impossible chance of it altering your DNA. So it goes into the fluid around the nucleus, which is called the cytoplasm. And in that cytoplasm, you have these things called ribosomes. And the mRNA acts as a template, basically a roadmap to say, hey, we need to make a protein. And that protein is the spike protein. Now, what happens is once that protein is made, that mRNA is degraded, it's gone. So that part of the vaccine is gone. Now what you're left is the spike protein. And what happens is two different things. One, parts of the protein are broken down by these enzymes that you have in your cell called proteasomes. So the spike protein is broken into smaller pieces and it's put on these receptors on the outside of your cell that are called MHC receptors. Now, most of your cells have a type called MHC type 1. These are also called HLA, human leukocyte antigens. And what these things do is they tell your immune system if something inside that cell is foreign or native. So your cell is constantly breaking down these proteins and putting them in this MHC1 receptor. Your immune cells will then come by that cell and read that little bit of amino acids or proteins in that MHC receptor. And it knows to recognize if that bit of protein is from your own cell or something else. So where this is really important is viruses, because viruses are not alive. They need to be inside your cells and use your cell machinery to replicate. So bits of viruses inside your cell can get broken down and put in this MHC1. And when your immune system cells come over and see, oh, this is not part of itself, it destroys that cell. It creates something called apoptosis, which means that the cell's DNA breaks down and no longer replicates, and the cellular machinery essentially is inactivated. This kills viruses. It also can kill things like cancer cells that are dividing out of control, because oftentimes these cancer cells have mutations to the point where the proteins in these MHC receptors are no longer recognized as self. And the cells in your body that are responsible for reading those MHC receptors and subsequently killing those cells are called cytotoxic T cells, or CD8 cells. That's one part of the immunity from this vaccine, is that it triggers these CD8 cells to say, oh man, a lot of these cells are infected with this foreign protein. It knows to recognize that foreign protein in the MHC receptor and then kill those cells, so that when you're infected with the actual virus, those T cells that were created while you got vaccinated, some of them, a lot of them die off, but some of them become what's called memory T cells, so that when you're infected with the same pathogen again, they're essentially woken up and divide rapidly to clear out the infection that's inside the cells. And why this is so important and why the mRNA vaccines are so clever is that antibodies don't work against viruses that are already inside your cells. They only work against viruses that are outside of the cell. So having a vaccine that can elicit both immunity that comes from killing already infected cells and antibody response is the most effective way to promote a robust immune response when actually challenged with the virus. Cool. So now that you understand that part of the way the vaccine works is that it destroys infected cells, this is part of the reason why you get arm pain and swelling and some of the commonly reported side effects from the vaccine is because those T cells are destroying the cells, and they're also creating inflammatory chemicals that are called cytokines that help bolster the immune response and bring more of the immune system cells to the area of vaccination. So the second part of how the mRNA vaccines work is after the spike protein is made, not all of it is degraded inside the cell. Some of it is transported to the outside of the cell, the outer surface of the cell membrane, and expressed on the outside of the cell. Then you have a second arm of your immune system that are what they call specialized antigen presenting cells, or APCs. Some of these can be things like, we call them dendritic cells, and they look like a sort of tentacle monster. And these cells are looking for foreign pathogens outside of the cell. So remember, the CD8 cells are looking for the ones inside your cell, and the dendritic cells and the antigen presenting cells are looking for things that are outside of the cell. So the dendritic cells will find these proteins that are expressed on the outside of the cell and give them to the other type of T helper cell, which are the CD4 T helper cells. And these T helper cells will then go over to another type of immune cell called a B cell. And B cells are the other way you produce immunity. So we all think about vaccine equals antibodies. Well, the way you get there is you have to activate these T helper cells through the use of the antigen presenting cells, which then present that foreign antigen to the B cells. And if the B cells have an antibody on their surface that recognizes that antigen, what it'll do is it'll start producing antibodies to that antigen. But those antibodies don't work particularly well. But some of these B cells will then travel into a lymph node. And what these lymph nodes are like a dojo. And if you start at the very basic level, these B cells that are in the dojo are constantly mutating themselves to see, can I make antibodies that better target this antigen or virus in this case? And the way it works is kind of like a lock and a key. So the key is the antibody, and the lock is that protein that's expressed by the virus, in this case, the spike protein. What you want to do is, even a kind of broken key will fit in the lock, but what you want to have is a perfect match so that that door opens easily. So what happens is, in that dojo of a lymph node, those B cells are mutating constantly to see, can we make a better key that fits this lock? The ones that get better at fitting the lock divide. Those that don't die. What ends up happening is, over time, and this is why you want to vaccinate, is because this process takes time. You develop a better key to the lock, or the best key to the lock. In this case, developing antibodies that are usually called IgG or IgA. And these antibodies are the ones that will bind to the virus and keep it from getting into the cells. And most importantly, in the studies, they didn't see something called antibody dependent enhancement, which is where antibodies actually don't inactivate a pathogen, but allow the pathogen to infect your immune system. It's kind of complicated. It's bad, but most importantly, it's not happening with these vaccines. So you don't have to worry about that. So this vaccine, because it's making the spike protein and expressing it on the cells, you're triggering that B cell pathway and you're producing antibodies as well. So it's working via two systems. And those B cells and that are producing the correct antibodies, the correct keys to the lock, two things happen to them. One, they'll form what's called a memory B cell. So some of them will essentially go into hibernation after vaccination so that if you're reinfected with the virus, they can rapidly divide, and now you already have the black belt antibodies against this virus. And then the second thing they form are what's called long-lived plasma cells. So these are the cells, the specialized B cells that are just pumping out those antibodies. So after you get the vaccine, you get a big response of these B cells, but a lot of them die off. Some of them go into hibernation, and that is your key to rapidly fighting the virus if you're ever exposed to it again. And also in a way that is not out of control. So it's not producing the cytokine storm that you see with actual SARS coronavirus infection. The other thing is the virus itself, if you're infected with it, actively down-regulates your ability to put those proteins in that MHC1. So the virus has these clever ways to keep itself from getting recognized. So when people think, I'll just get the disease and then I'll have immunity, the immunity that you get is unpredictable. And the older you are, the less likely you are to develop those specialized black belt B cells and those memory cytotoxic T cells that recognize, hey, this cell is infected, kill it. You're more likely to get that cytokine storm, which is what leads to the heart attacks and the strokes and organ damage that's killed so many people in this pandemic. An important thing to keep in mind is the timeline of all this, where a lot of people think that this vaccine is rushed, and that is the furthest from the truth. So the first step in developing this vaccine is you needed the genetic code of the virus, the sequence of the virus. And the sequence of the virus was released to the global health community and research community on January 10th, 2020. So once they have this, the mRNA technology and the platforms had already been developed. We have already been working to develop mRNA vaccines for other infectious diseases. So a lot of the technology that went into the vaccine was already there. A lot of vaccine development is money. So Operation Warp Speed helped take away a lot of the risk associated with developing vaccines. So typically vaccines are losing money in its development because it's a very long development time, and typically vaccines are relatively cheap. So there isn't usually a financial incentive for drug manufacturers to make vaccines. But with Operation Warp Speed and other similar programs that other countries ran, they gave a financial incentive to the vaccine companies by essentially promising to buy their vaccine ahead of time. So with the financial incentive there and the fact that this was a unprecedented global catastrophe, people got to work right away in developing it. But because the technology that mRNA vaccines was based on was already existing, it was simply a matter of finding the part of the virus genetic code that would be most likely to produce the immune response. Now, the hypothesis of using the spike protein, there was another SARS coronavirus pandemic in 2003 and 2004. And that particular coronavirus also uses a spike protein to attach to the ACE2 receptor, just like SARS-CoV-2, the virus that causes COVID-19. So we had lead time. We understood things about this virus beforehand. They're not the completely same spike protein, but similar enough that we said we should look at this target. If we can block its entry into the cells and make it a part of that the immune system recognizes, we could severely diminish infections and severe COVID, which is what the trials have shown, that people who are vaccinated are much, 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 much less likely to get severe COVID-19 if exposed to the virus. So the next part of the timeline that's important is, when did the vaccines that we have available now first go into people to be studied for safety and efficacy? And that is March 2020. So as we were starting to go into lockdowns, the vaccine was already going into participants to see how safe it is. And that number of participants steadily climbed as it went through the 3 phases of clinical trials. None of the clinical trial phases were skipped. All of the trial phases were done. They were just done in a way that was purposed and started very early in the pandemic. Nothing about this was rushed, and the data for it is solid. Between the 2 Pfizer and Moderna vaccines and their phase 3 trials, they had over 70,000 participants. 70,000. So when you have a participant size that large, you can usually tease out safety concerns because these people are being followed for months and are still being followed. Once a vaccine is available to the public, the research on it doesn't stop there. It goes into phase 4, which is the post-release, where you're still looking at vaccine side effects, efficacy, but now you're looking at it on a much larger scale. And now we have millions of doses at this point administered. And the safety data for it is absolutely outstanding. And this goes without saying. I know a lot of people are scared about allergic responses to the vaccine. I've had both doses at this point. If you have a history of allergic responses to vaccination, they have you wait 30 minutes after you get the vaccine to make sure that you don't have an allergic reaction. And the vaccine administration centers have medications to be able to respond to allergies to the vaccine. The important thing to keep in mind is, even if you had an allergy to another vaccine, the mRNA vaccines are so different component-wise to previously available vaccines, you likely will not be allergic to the mRNA vaccine. So just because you're allergic to something else does not at all mean you're going to be allergic to the mRNA vaccine. So moving on, how did I get to the point where I felt like I was ready to take this vaccine the first week it came out? And by the way, if you want to learn a little bit more about the vaccine and the differences between the Moderna and Pfizer vaccination, I gave a grand rounds back in December that goes into great detail about this. Now, keep in mind that the audience for the grand rounds was physicians, but you can see the video by clicking here, I guess. Hopefully the video just appeared and it didn't cut my head off. And you can learn a lot more about the differences between the Pfizer and Moderna vaccine. But anyway, how did I get to the decision where I was ready to get it? I looked at the phase 3 data and saw that the vaccine was very well tolerated, that they included people of demographics, gender, and age and different health disparities that actually represented the United States. So including a large amount of African Americans, a large amount of elderly, a large amount of people with medical comorbidities. They even included patients with well-controlled HIV in the studies. The main downside of the study is, like many studies, they excluded pregnant individuals, and they did not have people who were severely immunocompromised, such as people who have active cancer. Now, the American College of Obstetrics and Gynecology has come out and said that even without the data specifically on pregnant individuals are at a higher risk of developing severe COVID and should still get vaccinated. And as someone who understands the mechanism of the vaccine, I also believe it's safe to vaccinate pregnant individuals for this very reason. I would also say the same about immunocompromised individuals. Just because it wasn't studied, these are some of the individuals who are most likely to die if they get infected with COVID-19 and would not hesitate to give them the vaccine because of its safety profile and its efficacy. So knowing the research that was conducted and the fact that none of the safety steps were cut in developing the vaccine is how I felt confident enough to roll up my sleeve and get the vaccine the very first week it was available. And I have absolutely no regrets about that, and was proud to be part of this physician and healthcare community that took the vaccine and documented it to other people to show, look, I believe in this enough, and I believe in its safety and its efficacy that I'm willing to get it. And I think that says a lot. If there was a lot of hesitation in the healthcare community, and unfortunately there are still people that have not gotten the vaccine in the healthcare community, I don't think the vaccine uptake in the general public would be warranted at all. So anyway, this is getting a bit long. I just want to say, check out the Grand Rounds video if you're interested in learning more about the vaccine. And I even talk about the AstraZeneca vaccine for all those in the UK in that video. And keep it here on the Relaxing Sounds of Infection for more information about coronavirus vaccines and the coronavirus pandemic. Oh, did I mention you should absolutely positively check out drdansguide.com? That's drdansguide.com. I hope a website is popping up here. Otherwise, it's going to look really, really weird.